Thank you. It's a real honor to be here. I'd like to tell you very quickly a pure classical genetics story that began for me about 17 years ago. At that time, we were speaking of the phenotype gap that exists in mammals. In other words, we were aware by that time of all the protein encoding genes, but we didn't really have enough phenotype to understand what most of those genes did, and that's a problem that persists today. To help bridge the phenotype gap, we began inducing phenotype at random with a chemical mutagen, ENU, in mice. And at the time, it was a blind process. We didn't know really how much mutation we were inducing. We only knew that we did create mice that looked unusual, as I think you can see. We also learned from this exercise, making and solving more than 100 phenotypes by positional cloning, that if you saw a phenotype, it almost always resulted from a change in coding sense. This was a difficult process in the beginning, but it became easier, first with the annotated mouse genome sequence, which was published in 2002, then with methods for sequencing whole mammalian genomes and eventually even whole mammalian exomes. But genetic mapping remained a bottleneck in the whole procedure. The process to be followed once one saw a phenotype was to make a homozygous stock, then to outcross to another strain that had markers that could be followed, establish a critical region by backcrossing and phenotyping and genotyping F2 animals. And then finally, one could sequence the whole genome or whole exome to find where the mutation was. But this process of stock establishment and outcrossing and backcrossing took much too long. And suffice it to say that people saw many more phenotypes and named many more than they could ever solve. So we set about to make a new process for instant identification of causative mutations. That's now a reality in our laboratory, and in brief, the way we do it is to mutagenize, as we always did. However, what's different is that we whole exome sequence every G1 mouse that results from a mutagenized father, and in that way, we find all of the mutations that can be transmitted into a pedigree to G2 and G3 descendants. We began this work now about 3.5 years ago, and we can say with confidence that the average number of mutations that change coding sequence in each G1 mouse is about 60. And this is based on now more than 3,000 exomes sequenced. If we see that there are more than 30 mutations in a G1 mouse, we consider that that mouse is worthy of analysis, and we move forward to breed it, and in the meantime we order a set of AmpliSeq primers that let us genotype all of the mutant sites across the genome just within that pedigree. We typically make not just 2, but now 10 or 12 G2 mice, and we backcross them all to the G1 to get as many as 50 G3 animals in which we can analyze phenotype. All of the G2 and G3 mice are then sequenced using the AmpliSeq primers to determine their zygosity for all the mutations in that pedigree in every mouse. This is done with ion torrent sequencing, which is not nearly as fast in throughput as Illumina sequencing, but it's very nimble and one can analyze 2 or 3 pedigrees per chip and keep ahead of the process in that way. Typically, one can visualize all of the mutations, and I've made a kind of a chart here with mutations on the vertical axis and individual G3 mice on the top axis. I've color-coded the zygosities, so you see for a particular mutation, one has, for this mutation, for example, a certain number of homozygous variants in blue, heterozygotes in green, and reference allele homozygotes in yellow. And all this information is maintained in the computer until the mice begin being phenotyped, and when phenotypes are uploaded, an immediate search for the cause of phenotype begins. Just to show you that there's no ambiguity in terms of assigning genotype, these would be typical numbers: 514 mutant allele, 0 reference allele calls, the converse for a homozygous reference allele mouse, and this would be a typical heterozygote. And to show you even more clearly, there are really just three populations of genotypic calls: reference allele, heterozygote, or variant calls, as we refer to them. And there are big gaps in between. You have pretty good certainty about the genotypic calls that are made. The phenotyping pipeline that we presently use is illustrated here. We search first for dystonias, other movement disorders, then for innate immune competence, then for adaptive immune competence. We also screen for insulin production and also for glucose tolerance. We look for allergy. We do a bone density screen, blood pressure, and heart rate. We then look at a screen for the ability to repair the epithelial lining after it's damaged in the intestinal tract with dextran sodium sulfate. And then the pipeline is split into various sub-pipelines. We look for the ability to reject a syngeneic melanoma. We look also at addiction-related behavior, at the eyes of the mouse, at circadian rhythms, at fear, and we have a separate screen for autism that involves pedigrees all its own. On average, each mouse is assayed in 89 different ways. So a fairly broad net is cast to catch phenotype. Now, it's a very complicated matter and something that we've paid a great deal of attention to. You may be wondering, how much damage have we done to the genome in looking at all the mice we've looked at? And I'll give you the numbers. And I'll ask you just to take my word for it that we're able to calculate damage fairly accurately now, and to establish a range of genes that have been either destroyed or damaged enough that we would see a phenotype if one can result from that particular gene. The range of saturation we've achieved is somewhere between 26.7 and 31.1% of all genes damaged or destroyed and examined 3 or more times in the homozygous mutant state. To keep track of all of the mutagenesis that we've done, we had to make still other software. We wrote a program called Linkage Explorer, which lets us search by gene or by particular phenotypic screen or by any collection of mice. We also can specify the p-value of associations between mutation and genotype, and we can specify the size of the pedigree, the number of homozygous variants, and other factors, and then we can search for variants. Let's take an example. We happen to be interested, among other things, in the performance of the NLRP3 inflammasome, which I'm sure you're all familiar with. And here we've looked for pedigrees that are greater than 15 G3 mice, and with a p-value of 0.05 to establish linkage between mutation and phenotype. And if we hit return in the real program, we immediately get a list of genes that have been mutated and the number of alleles in particular genes, and some would be familiar and some not. One that was new to me was a mutation in NEC7. It was said to be a nonsense codon, judged probably null in its effect on the protein. And if we scroll to the right, we see the number of reference heterozygote and homozygote mice in the pedigree. And the computer has performed an analysis using an additive or a recessive, or still farther right, a dominant model of inheritance. Where the numbers are red, you're seeing p-values here. The numbers exceed the threshold that was input in this search. If you click on one of these numbers, then you immediately get a Manhattan plot showing linkage of every mutation in the pedigree to the phenotype that we've specified. And if you were to click on this particular point, you would see that that's NEC7. Knowing nothing about NEC7, it's convenient that the computer already has gathered information about it, and one can see, for example, a smart model showing where the mutation is, what it does to the protein. You can also look at a gene model, and you find that this is an exonic mutation, not likely to affect splicing. If it had, it would tell you what it was likely to do to the protein. If you right-click on this top point, then you get the phenotypic data displayed. These would be homozygotes in terms of IL-1 beta production by macrophages, heterozygotes, and reference allele homozygotes from the pedigree. And it's this declining relationship that gave strong indication of an additive inheritance of the mutation. NEC7 was previously known as part of the mitotic apparatus. It bound to the spindle, and this is work that we've all published, and what we can say with confidence now is that not only does it have a role in mitosis, but also during interphase it binds tightly to the leucine-rich repeats of NLRP3, and it's necessary for efficient assembly of the inflammasome, and then all that the inflammasome does, it cleaves, of course, caspase-1 and generates ultimately IL-1 beta, IL-18, and IL-33. So that's how we're able to rapidly go through the genome and analyze mutations and their effects. As soon as we see a phenotype, we know the mutational cause with pretty good confidence, that we always verify finally by CRISPR-Cas9 targeting. As we do this, we tend to hit the same genes over and over again, and as that happens, the computer realizes this and it generates super pedigrees combining all the pedigrees that have mutations in a particular gene and allowing us to sum the effects of the mutations in one gene or another. At present, there are 17,796 genes that have more than one mutation, and that's 71.2% of all genes. With multiple alleles, of course, your confidence about cause and effect increases. Here is a super pedigree for the sake of example. This is a particular mutation that was seen again and again, and the reason it was seen many times was that actually it's a contaminant in the Jackson laboratory stock, which has since washed out, but that does happen. In this case, there were 16 pedigrees, and you see there are hundreds of mutations, but only this one gives the phenotype, and because we've observed it with many, many mice, we now have an extremely strong p-value. If you right-click on that point, you see that the homozygotes show in this particular phenotypic analysis fewer CD8 positive T cells than heterozygotes or reference allele mice. This would have been very hard to positionally clone because of the overlap between heterozygote and homozygous variant groups, phenotypically speaking, and yet there's no ambiguity about it. This is highly significant, and this is the causative mutation, and it just fell into our hands without any particular work. Now, I'll tell you one biological story very quickly, how we found a new cause of diabetes in the mouse that I think is very important for the human. It happened that there were mice with growth retardation in a particular pedigree, and this was seized on by Zhao Zhang, a postdoc in the lab, who eventually made a tabulation of the weights of all these mice over time, and you see that they never grow to more than about half of the littermate size, and we called the mutation in question teeny for that reason. The teeny mice then have a diminished body weight. Interestingly, they have a high IGF-1 concentration, and you'll understand the reason for that presently. We found in short order that they also were hyperglycemic, severely diabetic in fact, and they had also a high hemoglobin A1C. If challenged with insulin, they don't become hypoglycemic as normal mice do, and so they're simply very insulin resistant. They're also lipodystrophic, and you can see here the inguinal fat pads, and or do I have it backward? This might be, yes, I think this is inguinal and this is epididymal, and you have some attrition of brown fat as well. Correspondingly, like most lipodystrophic animals or people, they have a fatty liver, and the liver weight is more than twice what it should be for body size. Now, unlike in the old days, we didn't have to go tracking this down by positional cloning. We knew immediately what the causative mutation was with pretty good confidence. There was linkage actually to two co-localizing genes, one a chloride channel, one a putative E3 ubiquitin ligase. And the ubiquitin ligase had a null allele which made us favor it. Ultimately, we targeted that gene with CRISPR-Cas9, and when it was knocked out, we found a perfect phenocopy of the teeny phenotype with, again, low body weight, high glucose, high insulin, and also resistance to insulin. We suspected that perhaps this phenotype all resulted from the action of the gene in fat because of the lipodystrophy and the diabetes that we observed. So we did fat transplantation from wild-type animals into the knockout, and this can be done by mincing fat and injecting it subcutaneously. Then you get these large fat pads that are well vascularized. And remarkably, within about 2 weeks, that will completely correct the diabetes. So insulin declines, glucose, as you see here, is normalized. This is a sham transplanted animal in both of these cases. And the liver improves, it's no longer filled with fat. Everything is pretty much corrected in the mouse. We suspected, based on structure, that this protein, about which not a single paper had been published, would interact with cullen, and cutting away parts of KBTBD2, we found that it would, and these are the bands that document that. And we wondered then, naturally, what's the target of this E3 ligase? And we performed mass spectrometry on fat cells and found something very striking. At the very top of the list, we found that there was a 40-fold excess in epididymal white fat of P85 alpha, the regulatory subunit of PI3 kinase. And this made sense to us because this is, of course, a key protein in the insulin signaling pathway. In the absence of KBTBD2, there's accumulation of P85 alpha, so we presumed it might target P85 alpha, and this is the documentation of that excess of P85 alpha on a Western blot. We did a protein blot then to see whether KBTBD2 would physically interact with P85 alpha, and it appeared that it did. At least it was very high on the list of interactors. And we further did a pull-down assay with a GST-tagged P85 alpha, and again documented direct interaction with KBTBD2. By cutting away the protein again, we found that this time it was the C-terminal domain, which has Kelch domain repeats, that seemed to interact. So apparently the Kelch domains bind P85 alpha, the BTB domain binds cullen-3, and it's known that that fixes an E2 ligase, which then can add ubiquitins to P85 alpha. In vitro, we could also document that P85 alpha was ubiquitinated with K48 linkage ubiquitin by mixing all of the components. In other words, ubiquitin, P85 alpha, KBTBD2, and cullen-3. And removing any of them would decrease the reactivity, although there's always some in the background, as these are cell lysates expressing each component. I want you to notice that if there is an excess of P85 alpha, if we do a pull down with the insulin receptor substrate 1, then you find that the active subunit of PI3 kinase is not phosphorylated as it is under normal circumstances in the wild type. And it's this failure to phosphorylate the P110 subunit that interrupts insulin signaling. So in the normal situation, we would hypothesize P85 alpha is made, but much of it is degraded as a result of the action of KBTBD2, and only some of it participates in insulin signaling. On the other hand, if you knock out KBTBD2, P85 alpha accumulates to high levels, and as Lou Cantley has shown, when there's too much P85 alpha, then a sequestration complex forms and the insulin signal is interrupted. If all this is true, then we should be able to suppress the phenotype of KBTBD2 knockout mice by additionally knocking out P85 alpha. And that we did, targeting a second time with CRISPR-Cas9. And you see here that it did correct the phenotype. There's an increase in body weight to normal. There's a fall in glucose, a fall in insulin at the right, and also I think that there's correction of fat weight in the third panel. There we are. Also, insulin sensitivity is restored, as you see at the bottom. So I've shown you that this mutation principally affects fat. KBTBD2 is required for insulin sensitivity of fat. KBTBD2 brings about the degradation of P85 alpha in fat cells, and if we restrict the quantity of P85 alpha in fat, then KBTBD2 will maintain insulin sensitivity. Without it, there's severe diabetes. So I've told you today how this process of mutagenesis has led us to find a new component of the NLRP3 inflammasome, also a new component of the insulin signaling apparatus, and I can tell you there are many, many other mutations that shed light on immunologic and non-immunologic proteins. To this point, then, we've destroyed about 30% of all genes in the mouse while keeping them under observation in a phenotypic pipeline that has close to 90 measurements in each mouse. In all, we've set aside a total of 981 phenotypically significant alleles of 698 genes that affect things that interest us. And a lot of things interest us, as I showed you from the phenotypic pipeline, but we focus mainly in the area of immunity. Of all of those, we've made public 486 of them in 267 genes, and there are 495 and 450 genes that remain restricted. In some cases, we're not absolutely sure that they are true, so we don't make them public yet. In other cases, we know that they're true, but we're still looking at them mechanistically. To help us in deciding what's true and what's not, given the number of mutations we've experienced with, we developed a meta-analysis program called Candidate Explorer, and it's able to predict whether things are excellent, good, or potentially good candidates. The sum of all those categories means that there's an 80% chance that they'll verify with CRISPR-Cas9 testing. That's a lot better than I can do, I have to say, because humans like me have a tendency to try to hope that things are true even if they're really not, and to put things into the CRISPR pipeline, which tends to clog it up. I'm often asked, is this not reverse genetics that you're doing now? And the answer is no, it's really still forward genetics. It's hypothesis-free. I'd say in some ways it's superior to the approach of targeting genes individually one after another, because certainly it's very easy to just introduce a spray of mutations into the genome of mice, into the germline, with a chemical mutagen. The new bottleneck, of course, is establishing mechanism, and I would say, looking at the whole platform, we will be using it more in the future for disease suppression rather than for creating disease. In the old days, it took 5 years for us to track something down. Now it takes really no time at all. It used to be very difficult to map. Now it's minimal work for people who manage that part of the pipeline. One phenotype used to be solved in the whole lab in 5 years. Now typically 1 or 2 are seen and solved every day, which is about 3,000 times as fast as before. Our limitations are at the level of screening, with 400 G3 mice entering the pipeline and a maximum of 600 G3 mice possible in our group. It takes quite a number of people to do this. There are 70 people in all working on the pipeline, and I want especially to acknowledge He-Sin Shi and Zhao Zhang for solving, respectively, the NLRP3 and the diabetes mutations I showed you. Thanks again very much.